Assalamu alaikum. So we talked about peptides, and we talked about the formation of a peptide bond between two amino acids. Now we're going to talk about polypeptides and how they form a protein with a distinct, definite three-dimensional structure that gives this protein a certain function. So now proteins have different structures. Okay, each one of these proteins has its own unique structure in three dimensions. Okay, three-dimensional structure. So some people would say, well, if we have a protein or a polypeptide that has 100 amino acids, what are the possibilities of different structures that can be formed? And it's basically gazillions. That is, in other words, it's a lot of probabilities. Okay, but however, in fact, the number of structures that can be formed realistically is about one or two, because there are forces and there are rules that govern how proteins form a three-dimensional structure. Now, this three-dimensional structure that has a function, we call it a native conformation. Okay, so each protein has its own native conformation, a three-dimensional structure that has a function. Now, before I proceed, just want to I want you to pay attention to a huge news that was announced about 2 weeks ago. And this is something that scientists have been working on for like 50 years. They're trying to predict the structure of proteins in three dimensions, again, according to the amino acid sequence of these proteins. So they have been trying to do that, and finally there were reports published 2 weeks ago claiming that scientists have been successful. European scientists have been successful in creating an artificial intelligence system that all you have to do is put in the amino acid sequence of a protein, and it can predict with high accuracy the structure of this protein in three dimensions. And you would say, so what? Now, it is really important to know the structure of proteins, because once you do that, you would be able to determine its function and the structure of the abnormal protein that causes a certain disease. Medically speaking as well, it's very important to know protein structure, because once you know it, you would be able to design drugs that can inhibit or activate this protein. And you can treat a lot of diseases as a result of that. And the system is known as AlphaFold 2. So it's really huge news, success story for scientists everywhere. So when we talk about protein structure, we simplify it by dividing the complexity of protein structure into 4 levels. And these are known as a primary structure, a quaternary, a secondary structure, a tertiary structure, and a quaternary structure. So the primary structure, this is the simplest structural level of proteins. It's basically the sequence of amino acid residues. That is, you say amino acid number 1 is this, amino acid number 2 is that, amino acid number 3 is this, and so on. So you say it's glycine, phenylalanine, tryptophan, proline, whatever. So it's just a sequence of amino acids. Now it's almost like you have a book and you read it, and all you read is letters. Okay, so it's like T, H, E, I don't know, F, L, O, W, E, R, the flower, I, S. Okay, so just R, E, D, red, whatever. So the idea is that you're just stating the letters, okay? then these letters can form a secondary structure. Okay, so this is a higher level of protein structure, the secondary structure, where you have localized structures. So within a large structure, you have certain localized structures here and there, and these are organized structures. Now then you have a third level, a higher level, known as the tertiary structure. And the tertiary structure is basically the three-dimensional structure of a protein, okay, in space, that is. Okay, so again, you have a book, you have certain letters, primary structure. These can be organized into words, and the secondary structure, and then you have the tertiary structure. You have the structure of the whole book, how these letters are organized in 3 dimensions in a whole book. Okay, now then you have the quaternary structure. That's when you have different volumes of the same book. So like it's an encyclopedia. So you say that's book number 1, book number 2, book number 3. They all make up one encyclopedia. So a quaternary structure is basically when you have a protein that is made of multiple subunits or multiple polypeptides. Okay, and each polypeptide, we call it a protein subunit, that is part of a protein unit, a whole protein unit. All right, so primary structure is when you have the sequence of amino acids, and we start with the N-terminus as always, right? So that's amino acid number 1. Okay, amino acid number 2, notice that they are in the trans orientation. R1, R2 is right here, R3 is right there, and so on. So that's the primary structure. We talked about that. Then you have a secondary structure. That's where you have a part of a protein having an organized specific structure like an alpha helix, and we'll talk about that. And then this alpha helix is part of a 3-dimensional structure of one polypeptide. Okay, notice it has a specific 3-dimensional structure. That's the tertiary structure. Then you have this protein that is composed of multiple subunits, okay, multiple polypeptide subunits, and that's how it looks like in 3 dimensions, and this is the quaternary structure. So let's talk about the primary structure and its importance. Now, again, the primary structure is basically knowing or telling or stating the order of amino acids, and these amino acids are covalently linked by a peptide bond, right? So right here we have leucine, glycine, threonine, valine, arginine, aspartate, and histidine. That's the N-terminus, and this is the C-terminus. Now, the primary structure, knowing the primary structure is very important, because once you know the primary structure, you can predict, as like I said, you can predict the 3-dimensional structure of a protein. And you can also tell, or you might be able to tell, what the function of the protein is. Now, sometimes you can have different proteins having similar primary structures with some differences. Okay, so we call these protein isoforms, and usually they exist in different tissues, having similar functions, but just like isozymes. We'll talk about isozymes later on. So these are known as isoforms. Okay, different shapes or different proteins that are similar to each other. So an example is hemoglobin. Now, there are different hemoglobin polypeptide chains. We have the alpha chain, we have the beta chain, we have zeta, we have the gamma chain. Now, if you look at them, you notice that there are regions that are highly similar among the different polypeptide chains. So these are isoforms. Now, there are differences, and these differences are the reason that gives these proteins their specific characteristics, whether we're talking about subtle structural differences or functional differences as well, maybe regulatory differences and so on. Not only that, but if you compare proteins among different species, like this protein right here, GAT or GATA or GAT-A2, if you compare the primary structure of the human protein versus mouse, zebrafish, it's a type of fish that is highly studied in laboratories. You would notice that there are, again, homologous, we call them homologous sequences or homologous primary structures among these different proteins. Okay, so you can discover a protein, for example, in a mouse, and you can predict that this same protein exists in human cells as well. Okay, and if you compare them, you can see that you compare these to this protein in different organisms, mice versus humans, you would find that they are highly similar. But there are, of course, differences as well. Now, the importance of a primary structure of a protein can be exemplified in sickle cell anemia. So sickle cell anemia is basically a disease that is caused as a result of the production of a defective hemoglobin. Now, hemoglobin is a protein that is responsible for transporting oxygen in our blood. Okay, so it's really important. It's a very important protein. Now, what happens with sickle cell anemia is that there is one little change in the amino acid sequence of hemoglobin. At position number 6 of the protein, instead of having glutamate, which is a negatively charged amino acid, you have valine, which is an aliphatic non-polar amino acid. So this is a huge difference. Okay, so changing something that is charged into something that is nonpolar would disrupt the protein structure. It would change the protein structure. Now, what this mutation does is that it changes the protein structure in a way that the proteins cluster together. So you have, instead of having each individual hemoglobin molecule by itself, rather you have clustering of the hemoglobin molecules. So they form an array of hemoglobin molecules. Now what happens is that, when you change the structure of the protein molecule, and you have millions and millions of hemoglobin molecules in one single red blood cell, and you have this clumping of hemoglobin, of all of these hemoglobin molecules, that would change the structure of the red blood cells from these nice rounded oval cells into elongated cells. And these cells also cluster together, they clump together. Okay, so we're talking about a little change in one single amino acid, which results, by the way, from a point mutation. Just one nucleotide that becomes different. It changes the amino acid of just one amino acid of the hemoglobin protein. That results in clumping of the hemoglobin molecules. That changes the structure of the red blood cells, so they clump together, and they clog the blood vessels. Of course, again, that results in tissue and organ failure, because there would be no oxygen getting into tissues. Okay, so knowing the primary structure of proteins is very, very important. Now let's talk about the secondary structure right now. Now, a secondary structure is basically, again, an organized, localized organization of amino acids into a specific structure. Now, remember I said that the peptide bond itself is rigid, right? So it doesn't rotate because of this resonance, this alternating double bond. However, the bonds within the amino acid residue around the central carbon can rotate. And that's what gives a protein its structure, its defined structure. Okay? So Again, the bond between the alpha carbon and the amino nitrogen, as well as the bond between the alpha carbon and the carboxyl carbon, can rotate. Now that results in the formation of hydrogen bonds as a result of the peptide, the groups that make up the peptide bond. And these hydrogen bonds can give a defined structure, like I said. And there are five structures, and these are known as the alpha helix, beta pleated sheet, turns, loops, and we have coils as well. And we can group coils with loops as well in one group. The first secondary structure that we want to talk about is the alpha helix. Now the alpha helix is basically, it looks like a helical rod. Okay, so it looks like a spring sort of, and it intertwines, okay, in space. Now each per turn, that is, if you go from this point right here all the way to it completes one full turn, you have about 3.6 amino acids. That is 3 amino acids and part of the fourth amino acid per turn. Now the pitch of the helix, that is the distance between 2 points on top of each other, is about 5.4 angstrom, and the angstrom is 10 to the minus 10th meter. Okay, now this helical structure is very stable, and the reason why it is stable is because of the formation of these hydrogen bonds between the groups of the peptide bond, that is the carbonyl group and the amide group. Now they lie sort of like on top of each other, so you have the formation of this hydrogen bonding between these groups. And that what actually determines the structure of alpha helices and what stabilizes the structure of alpha helices as well. Now, notice that the R groups are extended outward. So they are not embedded to the inside, rather they are oriented outward. So if you look at the N-alpha helix from top, so you have a top view looking from here all the way down, you can see how the groups are organized. That's the backbone of the peptide right here, and you have the R groups extended outward. Now, not all amino acids exist in an alpha helix. There are amino acids that are not preferred to be part of an alpha helix. An example is glycine. Glycine is the smallest amino acid. It's too small that it does not support the hydrogen bonding, or doesn't have an R group, just a hydrogen. So it really it disrupts the smoothness of the alpha helix. Proline is another amino acid that actually what it does, it breaks alpha helices. Usually proline exists at the end of alpha helices because it breaks the structure. It's a rigid molecule, right? Because of the cycle, the ring that is part of it. And as a result of that, again, the smoothness, the smooth turn of the alpha helix is disrupted. In addition, proline cannot form hydrogen bonds. It doesn't have a hydrogen bond donor. Its nitrogen is involved in hydrogen the formation of a peptide bond, and it doesn't have hydrogen, so it cannot donate its hydrogen in hydrogen bonding. Okay, so it doesn't support it destabilizes the alpha helix because of that. Now, you can have amino acids that are charged, and usually if they have similar charges and the R groups are close to each other, that causes repulsion, okay? As well as amino acids with branches at the beta carbon. Remember, the central carbon is the alpha carbon, and this is the beta carbon. So whenever you have branches or branching at the beta carbon, that results in increasing repulsion between the different R groups. So the secondary structure is known as the beta pleated sheet. So basically what you have in here is a combination of beta strands. This is a beta strand right here. Here you have the central carbon, that's the N-terminus. So you have the first amino acid. Okay, and then you have the second amino acid. Again, notice the trans orientation. Here you have the R groups going downward, upward, downward, upward, downward, and so on. Okay, so that's a beta strand. It looks like a zigzag. It has a zigzag structure. It goes up and down and up and down and so on. Okay, now a beta sheet is basically when you have a combination of these beta strands together, okay, on top of each other or next to each other. And these are hydrogen bonded as well. Okay, so I have this strand right here, and you have the other strand right next to it, and you have this hydrogen bonding between, again, the groups that make up the peptide bond, the carbonyl and the amide. And that's how these beta sheets are organized, or that's how they look in 3 dimensions. Now notice that the beta sheet itself is bent, and that's why it's called pleated. Okay, so notice that it goes up and down and up and down. Okay, so that's how the beta sheet looks like. Now these beta sheets, you can have within a sheet you can have 4 or 5, or you can have even more than that. It depends on the protein. So you have the combination of these beta strands making up the beta pleated sheet. And again, they are separated, they are stabilized by hydrogen bonds between the 2 groups, and they can be organized as in parallel formation or anti-parallel formation. So the difference here is that the anti-parallel formations, like it's going from the N-terminus, and then here you have the end of the beta strand. Okay, then you have a turn, and you have again continuation of the beta strand, or another beta strand. Okay, and you have a third one. And notice that the orientation is anti-parallel. They go against, or they oppose each other in direction. Whereas in the parallel formation, you have these strands within a sheet going in the same direction. So okay, so right here, as you can see here in 3 dimensions, you can see how they go in the same direction. Okay, that's parallel. Now notice the hydrogen bonding pattern is different. Okay, whereas within the same amino acid, so this is one amino acid, and this is another amino acid, they form two hydrogen bonds between them. Okay, so any 2 amino acids can form 2 hydrogen bonds, whereas with the parallel formation, the one amino acid would form hydrogen bonds with 2 different amino acids that are separated by a third amino acid. Okay, so right here you have 1, 2, 3 amino acids, and you have the formation of these 2 hydrogen bonds with these 2 amino acids, and there's nothing here. Okay, now you can have within a sheet, you can have multiple beta strands with going in same or different orientation. So right here you have anti-parallel strands within the same sheet, and you have another one right here as well. So so basically you can have combinations of these strands. Now, similarly, we have amino acids that do exist in beta strands or beta sheets. They support the formation of beta sheets, like valine, threonine, isoleucine, the same ones that disrupt alpha helices. They exist in beta strands, as well as large aromatic amino acids like phenylalanine, tryptophan, and tyrosine. They also tend to be present in beta sheets. On the other hand, proline, again the troublemaker, it tends to disrupt beta strands. Again, why? Because it's rigid. It doesn't support this nice zigzag structure of beta strands. Okay. So the way they are drawn, these alpha helices and beta strands, to simplify structures, is that the alpha helix looks like more like a spring, or again a helical rod. On the other hand, you probably have noticed that beta strands are drawn as arrows, thick arrows. So you have these two beta strands going in anti-parallel orientation. Now, we have a third secondary structure that is known as a turn. And these turns are really small. They are compact. They are U-shaped secondary structures. They're also known as beta turn or hairpin bend. And they basically can, they're really important in forming the three-dimensional structure of proteins, because you have a protein having a globular structure, like it looks like a ball, it looks like a globe, okay, globular. So you have this globular structure because of the presence of turns. So you have, let's say, an alpha helix going upward, so then you have a turn, and the polypeptide continues downward, and so on. So you have these different turns that form this nice globular structure of proteins. And what's really interesting is that proline and glycine tend to be present in beta turns. The reason is that the proline creates the kink, it creates the turn, the sharp turn, because of its rigid structure. And glycine fits in as the third amino acid residue within a turn because it's really small. Okay, so the turn is really, again, it's compact. It's a small structure. So glycine fits really well within a turn because it allows the polypeptide to actually turn smoothly. So proline exists as a second residue in a turn, whereas glycine exists as a third residue. And again, notice that that turn is stabilized by hydrogen bonding within the peptide bonds, the groups that make up the peptide bond, the carbonyl and the amide. Then you have loops and coils. Now, these secondary structures are irregular. Now, they're not random in a sense that a protein would form different structures of loops every single time it's produced. Rather, but it's not a specific structure like an alpha helix or a beta strand. Okay, so a protein would form the same type of loop every single time it is produced, but a loop in one protein would have a different structure than a loop in another protein. Okay, so loops are flexible structures. They provide flexibility to proteins. They are usually found on the surface of molecules. They can be extended outward of a protein, and they may have important functional purposes, as we will see later on. And usually these amino acids within loops are not conserved, so you can have any types of amino acids within loops or making up loops. Okay, so you have, notice here you have three different examples. These are loops, and notice how loops are different from one protein to the other. Now, these secondary structures can be organized in a way that they form what is known as super secondary structures. And these are basically structures that are composed of multiple secondary structures. So it's not a single secondary structure, rather you have a combination of secondary structures. And there are two types of super secondary structures. One is known as a motif, and the other one is known as a domain. So we'll talk about motif first, and then we'll talk about domain later on after we finish the tertiary structure. So a motif is also known as a module. Okay, so basically it's a repetitive super secondary structure. And usually it's repeated and organized into large, it can be repeated and organized in larger motifs. So usually, again, it's small. It constitutes about 20 amino acids, but it can be really large as well in certain proteins like immunoglobulins, and we'll talk about that later on as an example of globular proteins. So what do we mean by a motif is a repetitive super secondary structure? If you look at the primary structure of a protein, you see that you have a stretch of amino acids that can form an alpha helix. And these amino acids are followed by a number of amino acids that form a loop. And these amino acids are followed by another group of amino acids that form another helix. Now, this results in the formation of what is known as a helix loop helix motif. Okay, so you have the secondary structures being consecutive to each other. They are repetitive. You have one after the other. These structures are not separated by any other structures, even random structures. So you have a helix, loop, helix. Okay, so you have one after the other. There is nothing in between these secondary structures. So this is a type of a motif, and that's what we mean by repetitive secondary structures. So an example again is helix, loop, helix. So you have a helix, loop, helix. And usually this motif is present in proteins that bind DNA. Okay. Basically two alpha helices that are connected by a loop. Or you can have what is known as a helix turn helix. So you have a helix followed by a turn followed by another helix. Okay, again, this type of motif is present in certain proteins, the ones that bind to DNA. But having a motif, by the way, does not necessarily mean that this protein has this particular function. So you can have this type of motifs, the helix loop helix or helix turn helix, in different types of proteins with different functions and different localizations and so on. So motifs do not specify functions. That's what I want to say. Now you can have a more complex motif in immunoglobulins. Immunoglobulins are antibodies. We talked about antibodies before. So these are basically proteins that can recognize antigens or foreign bodies, foreign objects, foreign molecules. Okay, so that's how an immunoglobulin looks like. Okay, this is a simple drawing. So it looks like the shape of the letter Y. This is the part that binds to antigens. Okay, and note the structures, the different secondary structures. So right here, you have this part right here is known as the immunoglobulin motif or fold. Okay, so it's a super secondary structure, and this is the part that recognizes antigens again. Now notice that here you have in the immunoglobulin fold, you have a lot of beta strands. You can see the different arrows, and connecting these different strands, you have these yellow structures or drawings right here. These are loops. So these strands are connected to each other via loops. Now this is a motif because what you have in here is repetitive secondary structures. You have a beta strand followed by a loop, followed by a beta strand, followed by a loop, followed by a beta strand, followed by a loop, and so on. So basically you have a complex of secondary structures that are organized in one particular space. So this is known as this is an example of a complex motif.